Hello everyone, I am S. Jnanapunarchana from 2nd UG Microbiology. Now I am going to talk about the transduction in bacteria. Transduction. What is transduction? Transduction is a process by which bacteriophages function as intermediates in the transfer of bacterial genetic information from one bacterium to another. Once the donor genetic material has been introduced into the recipient, it undergoes genetic recombination with a homologous region of recipient chromosome. Transduction are of two types. Before that, we have to know about the lytic cycle and the lysogenic cycle. In lytic cycle, the phage takes over the bacterium and break down the bacterial chromosome and directing its growth and reproductive mechanisms to produce progeny phages. Finally, the phage is released by the lysis of the host cell wall. In lysogenic cycle, the phage chromosome does not replicate. Instead, it integrates into host chromosome. Every time the host cell chromosome replicates, the integrated phage chromosome also replicates. In this cycle, phage does not lyse the host cell, but it infects the host cell. Types of transduction. Transduction are of two types. One is generalized transduction, another one is specialized transduction. Generalized transduction. In generalized transduction, any genes may be transferred between bacteria. In this type of transduction, at first, bacteriophage infects the donor cell and begins the lytic cycle. When virus enter into the bacterial cell, some enzyme synthesize the virus components such as genome, capsid, head, tail, etc. Then viral enzyme hydrolyzes the host cell DNA and into small fragments. During assembly of virus component to form progeny viruses, sometimes any of the fragments of donor DNA get incorporated into the virus capsid. Such abnormal bacteriophage, when infect a new cell, it can transfer this DNA into new bacteria. Since this donor DNA is not a viral DNA, it does not replicate inside the recipient bacteria but undergoes homologous recombination with the recipient cell's chromosomal DNA, forming recombinant cell. Example of generalized transduction includes Escherichia coli transduction by T4 bacteriophage. Now this is the diagrammatic representation of generalized transduction. From this diagram, we have been cleared about the generalized transduction. Next, specialized transduction. Specialized transduction can occur only through the lysogenic cycle. Here, only the specific part of the bacterial DNA is packed into the virus. It occurs when the prophage which gets inserted into the bacterial genome in the lysogenic cycle excises. When prophage excises from bacterial DNA, some parts of bacterial DNA which are flanked on both sides of the prophage are also excised. Here, the newly packed phage genome consists of both bacterial and viral genome. Later, when the virus with the recombinant genome infects a new bacterial cell, the bacterial gene also gets inserted into the host genome with the viral genome through lysogeny. The recipient cell now shows the newly acquired characteristics. Specialized transduction is commonly used for isolation and insertion of genes of choice. Example of specialized transduction includes Escherichia coli transduction by lambda phage. Hope you understand the transduction in bacteria. Thank you.